In this video, I would like to explain about post-translational modification. Post-translational modification. So, what is post-translational modification? After the synthesis of protein, any alteration in the amino acid sequence of that protein is known as post-translational modification. That is, it refers to any alteration in the amino acid sequence of the protein after its synthesis. It may involve the modification of amino acid side chain. It may involve the modification of the amino acid side chain or terminal amino or carboxyl group by covalent or enzymatic modification. That is, it involves the modification of the amino acid side chain, then terminal amino or carboxyl group by covalent or enzymatic modification. And generally, these modifications influence the structure, stability, activity, cellular localization or substrate specificity of the protein. So, this modification influence the structure, stability, activity, cellular localization or substance specificity of the protein. Now, the location of the post-translational modification is in the endoplasmic reticulum or Golgi bodies. That is, post-translational modifications are mainly occur in the endoplasmic reticulum of the cell, but sometimes it may continue in the Golgi bodies. It occur in the endoplasmic reticulum and Golgi bodies. Now we can discuss about post-translational processing. What are the processes to modify these proteins? After the synthesis is completed, proteins can be modified by various methods such as phosphorylation, glycosylation, then ADP ribosylation, hydroxylation and addition of other groups. These are the processes to modify the protein. First one is phosphorylation, then glycosylation, ADP ribosylation and hydroxylation, then addition of other groups. First one is proteolysis or proteolysis. As the newly synthesized protein is released in the lumen of the endoplasmic reticulum, the newly synthesized protein released in the lumen of the endoplasmic reticulum, the signal peptidase cleave the peptide sequence. The signal peptidase cleave the peptide sequence. Apart from that, some polypeptide sequence of the protein is also cleaved, resulting in the final sequence. For example, insulin is synthesized in the cells in its inactive form, which cannot perform its function. So, post-translational modification ensure the proper function, which involves the removal of the part of protein to convert into three-dimensional fully active form. Insulin is synthesized in the cells in its inactive form, so which cannot perform its function. So it undergo post-translational modification and remove a part of the protein to convert into three-dimensional and fully active form of the insulin. Next one is phosphorylation. It is the addition of one or more phosphate groups. Addition of one or more phosphate groups to the protein. And post-translational phosphorylation. Phosphorylation is one of the most common protein modification that occurs in animal cell. That is, it is mainly occurs in the animal cells. And majority of the phosphorylation occurs as a mechanism to regulate the biological activity of a protein. So most of the phosphorylation occurs as a mechanism to regulate the biological activity of the protein. These are the amino acids which undergo phosphorylation in animal cells. They are serine, tyrosine and threonine. Serine, tyrosine and threonine are the amino acids that are subjected to the phosphorylation in animal cells. Next one is glycosylation. It is the addition of carbohydrate molecules to the protein. It is the addition of the carbohydrate molecules to the protein or polypeptide chain and modifying it into glycoprotein. the addition of carbohydrate molecules to the polypeptide chain and modifying it into glycoproteins. So most of the proteins that are destined to become a part of the plasma membrane, or to be secreted from the cell have carbohydrate chains attached to the amide nitrogen of asparagine or the hydroxyl group of serine threonine. That is, they are O linked and these are N linked. And the N-glycosylation which occurs in the endoplasmic reticulum and O-glycosylation which occurs in the Golgi complex. So the carbohydrate chains are attached to the amide nitrogen of the asparagine or the hydroxyl group of the serine or threonine. So they are known as O-linked and the asparagine is known as N-linked, and the N-linked glycosylation occurs in the endoplasmic reticulum and O-linked glycosylation occurs in the Golgi complex. Next one is sulfation. Sulfation means the addition of sulfate molecules. And these modification mainly occur in the tyrosine residues. The sulfation mainly occurs in the tyrosine residues of proteins. And this tyrosine sulfation is catalyzed by the enzyme tyrosyl protein sulfotransferases. TPST, which are membrane associated enzymes of trans-Golgi network. They are the membrane associated enzymes of trans-Golgi network. There are two types of TPSTs. They are TPST1 and TPST2. Two types of transferase enzymes are TPST1 and TPST2. And the universal phosphate donor is 3-phosphoadenosyl 5-phosphosulfate. PSPA is the universal phosphate donor. That is 3-phosphoadenosyl 5-phosphosulfate. Next one is methylation. It is the transfer of one carbon methyl group. It is the transfer of one carbon methyl groups to nitrogen or oxygen to amino side chain increases the hydrophobicity of the meth protein. So the transfer of one carbon methyl group to the nitrogen or oxygen to amino acid side chain, which increases the hydrophobicity of the protein and can neutralize the negative amino acid charge when bound to carboxylic acids. So they neutralize the negative amino acid charge when it bound to the carboxylic acid. So methylation is mediated by the enzyme methyl transferase and S-adenosyl methionine. It is the primary methyl group donor. So the methyl group donor is S-adenosyl methionine and the enzyme which catalyzes the reaction is methyl transferase and the addition of one carbon methyl group to nitrogen or oxygen of the amino acid. Next one is hydroxylation. It is the addition of the hydroxy group to a protein amino acid. And it involves the conversion of CH group into COH group. And these hydroxylated amino acids are involved in the regulation of some important factors known as transcription factors. Among the 20 amino acids, proline and lysine are regulated by this method. Proline and lysine are the amino acids which are regulated by this method. Now we can discuss some other processes. They are summoylation. Summo means small ubiquitin related modifier protein. They are 100 amino acid residue proteins which binds to the target protein in the same way as ubiquitin. So, sumoylation by sumoproteins. They are small ubiquitin related modifier proteins. They are 100 amino acid residue proteins which bind to the target protein in the same way as ubiquitin. They also shows the transcription regulatory activity. And they help in the transport of the target protein from cytosol to the nucleus. They also help in the transport of the target protein from cytosol to the nucleus. Next one is disulfide bond formation. Which stabilizes the protein structure and involved in redox processes. So the disulfide bond formation stabilizes the protein structure and they involved in the redox processes. The next processes are lipidylation, acetylation, prenylation, etc. So after the synthesis is completed, the proteins can be modified by various methods such as phosphorylation, glycosylation, ADP ribosylation, hydroxylation and addition of many other groups. These are about translation modification, that is post-translational modification. Now coming to the significance of the post-translational modification. So, proteins are synthesized by ribosome translating mRNA. Proteins are synthesized by the ribosome translating mRNA into polypeptide chains, which then may undergo the modification to form mature protein product. So, from the mRNA, the proteins are synthesized as a polypeptide chain, and these undergo modification to form the mature protein product. Now, coming to its significance, they aid in proper protein folding. So, they help in the proper protein folding. For example, a few lectin molecules called calnexin, which binds to glycosylated proteins and assists in its folding. So, the lectin molecules called calnexin binds to the glycosylated proteins and assists in its folding. Next one is, it confers stability to the protein. So, it gives the stability to the protein. For example, glycosylation can modify the stability of the protein by increasing protein half-life. Glycosylation can modify the stability of the protein by increasing the protein half-life. Next one is, it protects the protein. Against proteolytic cleavage by blocking the cleavage site. So, it protects the protein against cleavage by proteolytic enzyme by blocking the cleavage sites. Next one is protein sorting or translocation. If phosphorylated mannose residues are present in the protein, it always goes to lysosome. So, protein sorting and translocation are also with the help of this post-translational modification. The next one is, it regulates protein activity and function. Phosphorylation of protein is a reversible post-translational modification which activate the protein. Next one is the acetylation, which regulates many diverse functions including DNA recognition, protein-protein interaction and protein stability. So, the acetylation regulates DNA recognition, protein-protein interaction and protein stability. Similarly, it influences many aspects of cellular homeostasis. And post-translational modification are important components of cell signaling. Post-translational modifications are important components in the cell signaling. And it also increases the diversity and complexity in the proteome. So, the post-translational modification increases the diversity and complexity in the proteome. These are all about the post-translational modification and its significance. Thank you.